Great.
So, well, welcome everybody to the COVID-19 weekly seminar series. And today's format of bringing UCLA expertise together around a topic is one of the goals of our seminar series. So I'm really delighted to introduce Paul Ayoub and Melody Lee, who are both going to be talking about the advances they've made in pseudotyping for SARS-CoV-2. So Paul graduated from UC Berkeley with a degree in molecular and cellular biology before starting his graduate degree at UCLA in Dan Cohn's lab. He is currently primarily working on developing lineage-specific lentiviral vectors for the treatment of primary immunodeficiencies with hematopoietic stem cell gene therapy. But he's also been really eager to use his expertise in other areas, and he's actually been working with my lab over about a year or so now as part of a collaboration on looking at gene therapy for cystic fibrosis. And so I'm not really that surprised that Paul is also using his expertise now for COVID-19. And I do have to say that Paul is one of the most impressive graduate students I have ever worked with. He's got tremendous energy and he's really got amazing ideas. And when the pandemic struck, he conceived of this particular project with his mentor, Dan Cohn. But really, as Dan told me, all the design and the planning of studies was really his own work, which I think is really impressive. And one other thing I wanted to mention about Paul is that he became engaged to be married this year as well. So he's having a pretty landmark year. And so the title of his talk is Improved Spike Glycoproteins for Pseudotyping Lentiviral Introductions. But I'm also going to introduce Melody now so as not to interrupt the flow of the talks. And I have actually introduced Melody before. As you know, she's my co-host for the seminar series. And she previously gave a fantastic talk on coronaviruses. So I promised her that I would keep this introduction brief, but I want you to know that Melody is an assistant professor in MIMG, that she received her Bachelor of Science in Biological Sciences from the University of California, Irvine in 2006, and her PhD in microbiology from the University of Washington, Seattle in 2011. And she subsequently completed her postdoctoral work at Rockefeller University. She established her own laboratory at UCLA in 2017 and has already been incredibly successful. And her talk today is titled Pseudotyping Vesicular Stomatitis Virus VSV Particles with SARS-CoV-2 Spike Protein. So now, Paul, I'm going to hand it over to you as our first speaker.
Well, thank you, Bridget. Such an incredible introduction. All right. Well, let me get right into it. So I will be speaking to all of you today about my project in which we helped improve spike glycoproteins for pseudotyping lentiviral vectors. Perfect. So we generated a lentiviral vector that is capable of achieving a high titer, specifically in ACE2 expressing cells. And this was in the hopes of expanding research capabilities of SARS-CoV-2. And so to describe how we did this, for my presentation, I'll first discuss the background of the spike glycoprotein. And then second, I will get into our project objectives before going into description of glycoprotein variants, as well as our methodology. And then finally, I'll have our data on our spike pseudotype vectors, and then of course, conclude this presentation. And so as many of you know, COVID-19 virions are surrounded by a lipid bilayer in which these spike proteins protrude. So in short, the spike glycoprotein facilitates viral entry into target cells after it engages with the cell surface receptor angiotensin converting enzyme 2, or ACE2. And so this entry into target cells is mediated by two subunits within the spike protein. You have the S1 subunit, which mediates attachment to the ACE2, and then you have the S2 subunit, which mediates fusion into the cell with the help of various proteases. And so on the right-hand side here, I've depicted a snapshot of the SARS-CoV-2 viral entry. And so essentially the receptor binding domain, or the RBD domain within the S1 subunit, will engage with the ACE2 on the cell surface and lead to the priming or the cleavage of the spike protein at the S1-S2 junction shown here. And this is done by a protease, most notably the serine protease TMPRSS2. And so after protein priming, spike will then undergo a conformational change, and this will allow the fusion peptide to bind to the cellular membrane. And so subsequent to this fusion, the virion will then release its genetic material, and then this will ultimately lead to viral replication and release. The overarching goal of this project is to generate an efficient third-generation HIV-1 lentiviral vector that's pseudotyped by the spike glycoprotein. And what I mean by pseudotyping here is that we can effectively replace the envelope protein of the virus with that of another virus to either limit or expand the target cell population, targeting capabilities of our vector. And so in our case, we are limiting our lentiviral vectors so that they can only infect ACE2 expressing cells. Many groups have shown with previous viral envelopes that modification of their cytoplasmic tail can increase their pseudotyping efficiency. And so as such, we hypothesized by that replacing the cytoplasmic tail of the spike protein with that of another virus, say influenza or murine leukemia virus, we can increase pseudotyping efficiency greater than that of wild-type spike. We hope to utilize this vector towards research for SARS-CoV-2, especially since lentiviruses are non-replicating and they can remove the need for BSL-3 laboratories and potentially allow for things such as high-throughput drug screens, neutralization studies, mouse studies, and many other experiments. And then finally, our best pseudotype vector will then be produced in large batches with a GFP reporter cassette and be made available to UCLA investigators for research purposes. Getting into the nitty-gritty details here, there were 3 main modifications that were used to generate our spike glycoprotein variants, and I'll explain each of them in further detail in the next few slides. But in short, we either codon optimized our entire protein, we made amino acid substitutions, or we modified the cytoplasmic tail of the spike protein. So first, we codon optimized the spike. And for those of you that are not familiar with that, codon optimization helps improve gene expression of our vectors. And this is done by biasing the sequence of codons so that they're translated by the most abundant tRNAs within a host organism. Now, our constructs contained one of 2 codon optimizations, either optimization through an algorithm of integrated DNA technologies, or IDT, or codon optimization as described in a Nature Communications paper that was published in March of 2020 by a group in Beijing, China. And so this optimization was termed O after the last name of the first author. Next, we modified our constructs to include an aspartic acid to glycine substitution at the 614th amino acid position of the spike protein. This mutation, which many of you may be familiar of, mimics the D614G SARS-CoV-2 variant that has rapidly become the dominant form around the world. And our hypothesis was that potentially this mutant could help increase our pseudotyping efficiency in ACE2 expressing cells. And then finally, we have our cytoplasmic tail modifications. And I'll explain each in detail in the coming slides. But in short, we have our wild-type cytoplasmic tail shown here, and it was either mutated, termed ALAYT, or replaced with the cytoplasmic tails of influenza or murine leukemia virus, termed HA and MLV, respectively. The first tail, termed ALAYT, contains 2 mutations within the 5 most C-terminal residues of the wild-type spike cytoplasmic tail. And this mutant improves surface expression of the 2003 SARS coronavirus, shown here on the right-hand side. And that's done by eliminating the ER retention signal. And so given the sequence similarity between the 2003 SARS coronavirus and our current COVID-19, we went ahead and included this cytoplasmic tail modification. Then we have the HA tail, which replaces the spike CT with that of the influenza hemagglutinin proteins. And this has been previously also shown to improve pseudotyping efficiency in other viruses. But more recently, shown here at the bottom, the HA tail was also shown to successfully pseudotype lentiviral vectors with the SARS-CoV-2 glycoprotein. And so, of course, we went ahead and included this as well. And then finally, we have our MLV cytoplasmic tail. And so this replaces the spike cytoplasmic tail with that of the murine leukemia virus. And so previously, lentiviral vectors that were pseudotyped with envelopes such as GAL-V or RD114, shown here on the right-hand side, have been shown to have increased pseudotyping efficiency when their native cytoplasmic tails were replaced with that of MLV's cytoplasmic tail. So emulating these results in this yellow box here, we hope to achieve similar results with that of SARS-CoV-2. Now, putting all these changes together, so essentially all of our envelope proteins are driven by a CMV promoter. They also include a beta-globin intron as well as a beta-globin poly-A. And as I stated earlier, there were 3 main changes for every variant. We have our codon optimizations, either that of IDT or O. We have our inclusion of the D614G mutant. And then we also have our different cytoplasmic tail modifications, either wild-type spike, mutated ALAYT, or the influenza HA or MLV tail. And so several constructs were created to test how either all of these alone or in combination could affect our pseudotyping efficiency. So all the constructs were also compared to a VSVG glycoprotein control. and this is shown at the bottom. And the reason for that is VSPG has a broad tropism, so it's generally seen as the standard of comparison for any new envelope proteins. A little bit of overview of our methods. So we began by packaging the spike variants in HEC293 T cells. And this was with plasmids encoding for third generation lentiviruses, as well as a plasmid encoding for a GFP transgene. And then finally, of course, a plasmid encoding for our spike glycoprotein envelope. Next, we transduced our lentiviral vectors that were generated here into either ACE2 expressing 293Ts or HEC293Ts, which do not express ACE2, to assess the specificity of our pseudotype lentiviral vectors. After transduction, we measured GFP percentage and GFP MFI, and this was done by flow cytometry to assess expression of the transduced cells. We also measured TUs per mil by digital droplet PCR, and this was to determine the efficiency of particle formation. We also measured P24 particles, lentiviral particles, to determine the efficiency of particle formation. And then finally, we plan on measuring the spike protein via an ELISA. And this is to assess the relative amount of spike protein to total lentiviral particles. And we really just want to quantify here the bulk particles or those with an HIV capsid, but no spike envelope protein. Moving into our experimental design of the cell transduction, we plated 100,000 cells, either ACE293 T cells or HEC293 T cells in a 6-well plate. And then 24 hours after plating, the cells were transduced with a 1:10 dilution of raw viral supernatant. And then 3 days post-transduction, they were harvested. And then after harvest, half the cells were extracted for their genomic DNA. And so this was used to assess viral titer by measuring GFP integrants through a digital droplet PCR. And then the other half of cells were assessed for their percentage of GFP expression and GFP MFI, and this was done through flow cytometry. So taking a look at our flow data here, this is our flow gating strategy. So all cells were gated upon on a non-transduced control. And then of course our doublets were removed. And then after that, we went ahead and gated upon our GFP positive population using this non-transduced control. So taking a dive into our functionality and expression data, this is 3 days post-transduction of the first set of pseudotyped lentiviral vectors. So these vectors utilize a UBC M-citrine transgene cassette. And so getting into a little bit of depth here about our graphs. So on orange, our bars represent the ACE293 T cells, whereas in blue, the bars represent our off-target HEC293 T cells. And so moving from left to right, we have our non-transduced control, our VSVG control, and then we have our IDT codon optimizations alongside their relevant modifications. And then we have our O codon optimizations alongside their relevant modifications. So the first thing I want to note here is that our spike pseudotypes do not infect our off-target cell line, the HEC293 T cells. So you can see here that they do, however, specifically infect our ACE2 expressing cell line. And there are 3 variants I want to draw your attention to that increase the pseudotyping efficiency over wild-type spike in the ACE293 T cells. We have our HA tail modification, and then we also have our D614G mutations, both the IDT version as well as the O codon optimization version. Interestingly, however, the variants that included the MLV tail seem to impede our pseudotyping efficiency, as you can see both in the IDT and O codon optimized versions. And so they were therefore not used in subsequent experiments. And so taking the increase in efficiency here for our HA tail as well as our D614G tail, the next question became, could we combine these 2 modifications and actually obtain an additive or synergistic effect on pseudotyping efficiency over their sole counterparts? So we generated 3 new variants shown here, and we went ahead and packaged each of these variants, these combined variants, head-to-head in 293Ts alongside their relevant controls, as well as the previous spike pseudotypes that I showed in the graph prior, in the slide prior. And after packaging and harvesting our virus, we then repeated transduction experiments and assessed GFP expression via flow cytometry. So here's our flow gating strategy once again, gating upon all cells, selecting out our singlets, and then gating upon our GFP positive population. And so again, shown here is our functionality and expression data 3 days post-transduction, but this time with the new set of pseudotyped vectors. What I do want to note is that these pseudotyped vectors actually contain an MND EGFP transgene cassette. And why I think that's important is because the MND EGFP transgene cassette is actually a weaker promoter and a weaker expressing GFP than that of the UBCM citrine. As you can see with the GFP percentages shown here for the HA and D614G mutants that were lower than their previous experimental the previous experiment that we ran. And so like before, the orange bars represent our ACE293T transduced cells, whereas the blue bars represent our HEC293T cells, the off-target cell line. Again, you can see our vector shows specificity to ACE2 expressing cells. What I do want to draw your attention to is on this far right-hand side here, the combination of the OD614GHA mutant, or modification, excuse me, actually reaches a nearly 55% GFP expression using the 1:10 dilution of raw viral supernatant in these ACE293T cells. And shockingly, that's about 10-fold greater than its D614G cell counterpart. I would also like to note that all these constructs had similar levels of P24 value with a percent CV of about 5.1. And so after we assessed this functionality and expression and narrowed down our lead candidate here, the OD614GHA, the next goal is to see, can we quantify the infectious particles of each of our variants? And so we went ahead and measured the transducing units per mil to assess the infectivity of our pseudotyped vectors. So to do so, as I stated earlier, genomic DNA was extracted 3 days post-transduction, and the GFP integrants were then quantified by a digital droplet PCR. So the graph on this slide illustrates our viral titer here, but on a log scale. And so as confirmed by our flow cytometry, as well as the titer data, you can see here in blue that our spike pseudotypes do not infect our off-target HEC293T cell line. And that's just given the fact that their viral titer is similar to the levels of the non-transduced control. In ACE2 expressing cells, however, we do get specificity. Once again, I want to draw your attention to our OD614GHA mutant. And this achieves an impressive titer of about the mid-10 to the 7 range, which is a log lower than the VSVG control and about 3 logs higher than the wild-type spike IDT counterpart, which achieves a titer of about the mid-10 to the 4 range. And so in conclusion, we have generated a spike pseudotyped lentiviral vector that's capable of infecting ACE2 expressing cells with minimal off-target specificity. Our combined variant, the OD614GHA, achieves a titer that's 3 logs greater than its wild-type counterpart, while also retaining specificity to the ACE2 expressing cells. And our vector hopefully can remove the need for BSL-3 laboratories, given that lentiviruses are non-replicating, and potentially expand the research for SARS-CoV-2 so that we could open up to experiments such as high-throughput drug screens, neutralization studies, and other therapeutic aspects towards SARS-CoV-2. We are still in the process of quantifying the spike protein in our viral supernatants to rule out these bulk particles. So unfortunately, we are waiting on that data. But our spike, fortunately, our spike OD614GHA variant will be packaged alongside an MND EGFP transgene cassette and be made available to UCLA investigators for research purposes in a month's time. And with that, I would like to thank, of course, the Cohn Lab, specifically my mentor, Donald Cohn, for giving me such a great opportunity, and many others alongside this one, as well as Jason Quintos for helping me undergo these experiments, and Roger Hollis for helping me a lot with experimental design and moving forward with this project. I would like to thank the UCLA Virology Corps for their help with the P24, as well as a lot of investigators at UCLA who have helped me guide this project along, shown here. And then, of course, the UCLA Flow Cytometry Corps. And yeah, with that, I think, Melody, on to you.
Great, thank you so much, Paul, and very nice background to the pseudovirus. So I don't have to get into that in my talk. So let me just share. Can everyone see my screen?
Yes.
Yes.
Great. Move it over here. Okay. Awesome. Well, thank you, Paul, for such a great presentation. And, you know, it's awesome, all the different optimizations you've done. And I guess just a disclaimer, in my talk, we are really trying to set up a system with a minimal amount of optimization. So any suggestions for how to make the system even better will be great. And thank you to Bridget for, you know, the introduction second time around. And happy to be back today. And I'm going to talk about another virus today, vesicular stomatitis virus, pseudotyping system. So, okay, it's not going. How about now? Okay, so before I get into our system and the preliminary data, I really want to first thank everyone that's involved in this collaboration. This work cannot be possible without really the generosity of a lot of different people at different institutes. So. The goal of this is really to provide VSV pseudotype viruses to Robert Demisot at the high throughput screening facility so that he can screen for neutralizing antibodies in a convalescent patient sera. And Oliver Fragoso, a colleague of mine in MIMG, has been doing the experiments with me, so he's my partner in crime. We got the spike construct from James Valls from Scripps and the VSV delta G, so basically replication deficient VSV seed particles, that lacks the glycoprotein. We got these seed particles from Oscar Negrete from Sandia National Labs. And also really detailed pseudotyping protocol from Michael Lecco, who was a former postdoc in Vincent Munster's lab at Rocky Mountain Labs, and he's now setting up his own lab at Washington State University. And last but not least, we got the 293T overexpressing ACE2 cells from Peng Wang at USC and Lily Yang here at UCLA, also a colleague of mine in MIMG. And the work is also helped by my graduate student, Emily Yang, in my lab. And here are my funding sources for the different COVID projects we have that are ongoing in the lab. So I've shown this general life cycle slide for coronavirus in my very first talk, but today I'm going to focus on the very first step, which is the receptor mediated endocytosis and fusion step. And Paul has given a really nice introduction to how the S1 and S2 proteins of coronavirus engage with the receptor on the cell surface, which is angiotensin converting enzyme 2 ACE2. And I'm sure you all know that working with SARS or SARS-CoV-2 viruses is very cumbersome because you need to work with them in a BSL-3 facility. And working with live viruses also make it harder for adapting high throughput assays. And so the question we have was, can we establish can we establish a BSL-2 infection system to assay for factors that can modulate viral entry, specifically neutralizing antibodies and potential antiviral host proteins? And so we looked into the literature and found that one of the preferred systems in the field, in addition to the lentiviral pseudotyping system, is the vesicular stomatitis virus delta G, VSV delta G system. And this system produces particles that can incorporate envelope glycoproteins of multiple RNA viruses, including the Ebola glycoproteins. And so you can see here on the left here, that is adapted from a review article in Vaccines that shown on top is the VSV delta G genomic organization. And simply in this genome, the glycoprotein is replaced by reporter genes. So in this case, it's GFP and firefly luciferase. And for our system that I will talk about, it's M-cherry and Ranilla luciferase, so very similar. And you can see here that when you make the seed particles, meaning that you can transfect this construct VSV delta G genome, but with packaging, sort of like packaging constructs, but really plasmids that encode the individual proteins, viral proteins, are important for setting up the replication complex, because I want to point out that VSV is actually a monosegmented negative sense RNA virus. So once the RNA goes into the cell, it cannot be translated immediately unless it brings in its own replicase proteins. And so what you can do is you can co-transfect cells with all the different constructs that would express the replicase proteins, and that will allow production of seed particles, which we call, that are complemented with VSVG. So you will also give a VSVG plasmid. This seed particle will be able to infect cells that are transiently transfected with coronavirus spike protein of interest. And so that as new viruses come out, which you can see here on the bottom, that this new virus will be complemented with the coronavirus spike protein. And so when you take this virus and infect target cells, such as viral cells that have endogenous endogenous ACE2 expression, you can assay for viral entry by luciferase or fluorescence expression. And so we can imagine that if you pre-treat this pseudovirus with neutralizing antibody, or patient sera that have neutralizing antibodies, you can calculate a percentage inhibition conferred by the neutralizing antibody. So there are a few reasons why we initially decided to go with this system. One is that the pseudoviruses, these pseudoviruses grow at very high titers in a variety of cell lines, so they're very easy to work with. A replication time is extremely fast, or more that, I should say that more correctly, it's the expression of GFP, right? Because this virus is a single cycle virus. So because it doesn't have its glycoprotein, it cannot spread. And so GFP positive cells can be readily detected by 16 hours post-infection, as opposed to on average 48 hours for lentiviruses. And most importantly, for coronavirus spike pseudotyping, it's been shown in this study back in 2015, Nature Medicine, that lentiviral pseudoviruses have failed to reflect viral entry mediated by bat-derived coronavirus spikes. And it's not clear if that's the case for other coronavirus spikes, but in this study, what they found is that when they pseudotype lentivirus with the bat virus spike, it does not get into the cells through human ACE2 receptor. However, the live virus does get into the cells. And so there might be some discrepancies, and it's hand waving in the field when I talk to my coronavirologist friends why this is the case. And one potential explanation is that potentially lentiviruses, and again, maybe Dawn can comment on this more because I don't know very much about this, but lentiviruses are thought to incorporate less glycoproteins than coronaviruses will. So, and obviously what we saw, you know, with all the new studies coming out, we saw a recent study where they have already set up a VSV delta G pseudotype system with SARS-CoV-2 spike, and they found very nice dynamic range. So shown here on the left is basically a very similar system as I showed in last slide. And you know, seed viruses, seed particles are made with this VSV delta G construct, and they have a neon green and nanoluciferase reporters. And here is the spike plasmid that they've used for their transfection. And Paul has really nicely given the background on the cytoplasmic tail modification. And so the construct they use in this study, it has a 19 amino acid deletion in the C terminal end of the tail, which allows more efficient packaging. Our construct has an 18 amino acid deletion, so very similar to this construct. And what you can see on the right here is that you have very nice dynamic range comparing the viruses that are pseudotyped with the spike, which are the solid symbols, and versus the virus that don't have the spike, which are the clear circles. And these are actually HOH 7.5 cells are infected with these VSV pseudoviruses, which also have endogenous ACE2 expression. What is interesting to me is that it seems like the virus that has the neon green, or that's quantified by the neon green expression, has a smaller dynamic range than the nanoluciferase, so higher background. And I'll get to that in our data, where we think also the different reporters might be giving us different signal to noise ratio. And what is most important is that when they took plasma samples from patients, convalescent plasma samples, and they compared their two systems, so they also tested the HIV pseudotyping system, compared them side by side with the VSV delta G system, they plotted a correlation curve of the NT50, meaning that it's neutralization titer 50. So how much antibodies do you need to inhibit the virus by 50%, to neutralize the virus by 50%? And you can see here that on the y-axis, they also tested live SARS-CoV-2 virus. And both of these pseudovirus systems correlate very nicely with neutralization of the live COVID-2 virus. And so I think both are, excellent methods that can be adapted for high throughput assays, like what Paul alluded to earlier. And so what we have done, and this is the experimental flow through, is that on day one, we would see 293 T cells, which are, can be efficiently transfected. Then on day two, we would give the cells our SARS-CoV-2 spike construct, which I pointed out that it has 18 amino acid deletion in cytoplasmic tail for efficient packaging. Then on day three, we infect these transfected cells with our C particles, and generating the C particles is quite cumbersome because you have to co-transfect lots of different plasmids so that you can make a VSV virus. And so we got these C particles from our collaborator at Sandia National Lab. So we're very fortunate to have that, because then with a high titer C particles, we can just simply infect the cells that are transfected. And then a day later, we already harvest the VSV delta G pseudovirus that is, it has incorporated the SARS-CoV-2 spike protein. And maybe immediately or a few days down the line, we can then infect new target cells that have either endogenous or exogenous expression of ACE2 and measure luciferase or fluorescence for cell entry. And so this is what we found. So our pseudovirus has M-cherry or ranitidine. So we generated two different batches of pseudoviruses and we assayed them separately. So you can see here that on top is the bright field of the cells, and you can see cytopathic effects from the VSV delta G infection. And later on, when I show you, that correlates with high infectivity. And on the bottom is M-cherry channel. So that tells you how many pseudoparticles are there. So you can see that when there's no glycoprotein present in the cells that were infected with the C particles, the background is extremely low. I hope you can see the positive cells. And when we infected the cells are transfected with the SARS-CoV-2 spike, when we infected cells with the pseudoparticle that has a spike from SARS-CoV-2, you can see more cytopathic effects in the viral cells and also a lot more positive cells. And so it seems like our pseudovirus infects viral cells in a manner that is dependent on spike ACE2 interaction that is specific. We also have our control where the virus is that has VSVG glycoprotein, and you can see that expression is extremely robust. and efficient, and basically almost all the cells are already rounded up and exhibiting cytopathic effects. And this is, I just want to point out, this is using undiluted virus. And so next we looked at different dilutions, and what we found is that the pseudovirus that has the SARS-CoV-2 spike packaged at the 10 to the negative 2 dilution seems to be very similar to the level of positive cells that are infected with the 10 to the negative 4 dilution of VSVG containing pseudovirus. So I would say, and I think this is consistent with also what Paul showed earlier in one of the papers he cited, that definitely packaging with spike is less efficient than packaging with the VSVG glycoprotein. And so the last piece of data I want to show you is infection with our rhamnylluciferase expressing spike pseudovirus. What we found is that when we compared the luciferase activity, shown here counts per second, of increasing amount of pseudovirus in 293 T cells with ACE2 expression, so in the red here, the solid circles are infection with virus with spike, and the clear circle is virus without spike. You can see that the solid circle give us very nice high luciferase activity that is positively correlated with the amount of virus we put in. However, you can see that the dynamic range is not great, because the clear circle should represent the background, which is if you have no spike, these cells should not be infected. And we see about a 3 to 4-fold dynamic range. When we looked at cells that just regular 293 T cells that do not overexpress ACE2, we basically got similar levels of luciferase activity, which are similar to and maybe a little lower than the background we see in the ACE2 expressing 293 T cells, which is very consistent with the idea that, you know, at sort of at the level of the clear circle, the red circle, and also the 293 T, this is all just background infection. And what we saw when I looked into the other study I showed you earlier, where they compared lentivirus with the VSV pseudoparticles side by side, I was surprised to see that they also saw a pretty big, pretty high level of background. So you can see here that on the right, 293T overexpressing ACE2 has a very nice dynamic range. Definitely their dynamic range is nicer than ours. And I'll explain in the next slide, my last slide, what are the things that they did that we did not do that might improve the signal to noise ratio. However, I was surprised to see that in their 293 T cell infection, their background is quite high. And when the pseudovirus has spike incorporation, which is also the solid circles here, it seems like there's even more entry into the 293 T cells. So I'm puzzled by this, and I thought it's interesting that their curves are not more similar in the 293 T cells. So sort of the next steps that we want to do are things that we want to try to reduce the background. One thing that in the previous study I showed you that they did was they actually concentrated the VSV pseudovirus after they harvested it. And so supposedly it would remove any contaminating luciferase protein. The other things that we are in the process of doing or trying is that even though I washed the cells really, really well after I infected with the seed particles, because you can imagine that when I put the seed particles on the cells that have the spike protein, you don't want the new virus to come out to have still the VSVG glycoprotein, because that will give you the background. And so what I do is I wash the cells extremely well 5 times, which if you work with 293Ts, you know how hard that is. And so at the end, I would think that the washing would probably remove a lot of the contaminating VSVG particles. But clearly we still have a high background. And so some of the things that we're trying now is we have harvested the spike particles at a later time point, because you can imagine that if VSVG is very stable, after the C particles get into the transfected cells, the VSVG might stay on this on the cell surface or in the cell for a while, that perhaps the new particles that are being produced can still have VSVG sort of tag along with it when it buds out. And the other thing that the previous study I show you, in the JEM study that we were thinking about trying, is we can neutralize the contaminating VSVG particles with anti-VSVG supername prior to we infection of target cells. So maybe that will also decrease the background. Last thing we're thinking about is perhaps a shorter infection period could maximize the signal over background ratio, and because we've been doing infection for 24 hours. And obviously with this system, we can already detect reporter expression by 16 hours. So perhaps, you know, minimizing the infection will help with this. And so these are some of the things we're thinking about trying, but any suggestions will be great. And that's that. Thank you for your patience.
Great. So I'm going to open it up for questions and discussion right now.
So, Melody.
Hi, Hong.
Yeah, can you hear me? Okay, good.
Yeah, I can hear you.
So I think the thing you observed, actually, I think it's probably due to the fact that you have very high concentration of particles.
Mm-hmm.
The reason is for it because the VSV itself has a size about roughly about 500 to 50 to 100 nanometers in size.
Mm-hmm.
And with that kind of particle, nanoparticle, we call, and cells, any cells have a tendency to uptake because the cells mistake those as food. And in order to reduce this kind of background, I think one way to do that effectively just to decrease the concentration of your viral particle significantly.
Mm-hmm.
And what's the title of your particle?
You mean the one that is pseudotype with the spike or the VSVG?
Both, yeah, both. Because the bottom line is that particles in the range of 50 to 100 nanometers will be uptaken by the cells if the concentration is high.
Got it. So it's just like a passive uptake.
Yeah, you do not need any receptor recognition. The receptor only helps the virus to enhance the probability of being uptaken. But since you have a high concentration to begin with, then that enhancement doesn't mean you have 3 or 4 fold enhancement. That makes sense.
Yeah. Would chloroquine help that? Would it block that uptake pathway? Hate to sound like the president, but hydroxychloroquine.
It's macropinocytosis, so it should down, but it's going to block. It may also block uptake of the virus, right? Is uptake of the virus chloroquine sensitive because it's coming in a vesicle?
Yeah.
I don't know that.
I don't know.
For VSV, right? I mean, that's a good point. Han, do you know for VSV, is this endocytosis? I mean, I guess if it's endocytosis.
Yeah, you say, endocytic pathways.
Would be
cool.
Yeah. So,
I mean, you have a really good point. To answer your question, the VSV seed particles we get from our collaborator is, I mean, crazy high titer. I mean, that's why VSV is so great.
Yeah.
It's like 10 to the 8 focus forming units. And also it's replication defective, right? So let's say each M-cherry or GFP positive cell is one infectious virus or one particle. So it's quite high, and probably even when I wash them really well, there's just a lot.
Melody?
Yeah.
you have any way to assess the amount of spike that's being incorporated into viral particles in a pseudotype virus versus COVID-2? Because, I mean, this is avidity driven. So it's dependent on number, density, cooperativity, et cetera. Is there something that you can do to measure that?
Yeah, that's a great point. I think Paul is planning to do that in one of his future directions. We should definitely maybe compare our numbers. Maybe I should talk to you, Paul, and see if we can compare our particles side by side.
Oh, how are you planning to do it?
So mine are not actually uptaken in terms of, but it's just viral supernatant. So we're just assessing the amount of spike protein in viral supernatant, and it's done through an ELISA.
And then per P24 to normalize it to virion.
Yeah, and it's normalized to P24, exactly.
I'll tell you a better way. Just take some images. Use the CNSI instrument. You'll see all these spikes easily. You can count them.
Oh, that's okay. Is it easy to do that, Han?
Oh yeah, it should. If you don't mind, actually, if you actually others don't mind, I can show you a picture. You can see that very clearly. Yeah. If you don't mind, just let me see if I can find my file. Yeah, others can go ahead.
But yeah, Jeff, I guess we can also, I mean, in terms of quantifying the glycoprotein in the virus, we can probably concentrate the virus, right? And then maybe either do Western blot. I don't know if that's very quantitative. But maybe some kind of labeling. Yeah, I would say, yeah, probably EM labeling will be.
Yeah, can somebody let me to share the screen?
You should be able to, I think all participants can share screen if they need.
Okay, thank you so much.
Yes, it's opening.
Yeah, you can see, I don't know if you can see here on the screen.
Blow it up to full screen if you can.
Yeah, I did. I need to switch the screen. Where's the switching?
Maybe to blow it up bigger or
Yeah,
keep it
I just forgot how to switch this, but so you can see those are the VSV particles. These are the Wildtype. You can count it. You can count how many spikes you have.
That's really cool.
Right. So you can also tell it's pre-fusion and post-fusion. Yeah. You can you can even see this shape distribution.
So what do we need to give you?
Oh yeah, you can just give particles. I mean, give the viral particles. You can image it. You can see that. You can see this clearly, without any problem.
Yeah, it's really nice, and it's interesting because the density, the distribution of it, it's not even, right? I mean, I guess if it's just a cross section, maybe that's harder to say.
Well, it's not even. You can see there's a different kind of pattern clustering here. There's a different look at different perspective. This is around the spikes. You know, this tremor. There's a double tremors, triple. You can actually full clustering. The bottom line is you can see all these. You mentioned that you could have a mixture of the native plus your pseudotype, and then you should be able to tell also because they're different.
And the images you're showing here, these are VSVG glycoprotein.
These are the native wild type, yeah. Yeah.
The wild type. So obviously the coronavirus, the spike will be very different looking than the VSVG.
Yeah, it will be very different, yeah.
That would be cool. We should talk.
Yeah, you should use it. Great.
And maybe Paul can also give you some virus.
Oh, yeah, sure, yeah.
Count.
Yeah.
Great. It's awesome.
Melody, I have a question, or maybe for Paul as well. This may be a bit naive, but when I think about spike binding to ACE2, I think about other proteases that may be important in terms of cleavage and allowing for that entry. Is that important in your systems as well? And is it important in terms of different cell types?
Maybe Paul can go first.
All right. So I do know that cathepsin L and B, I think, are also important proteases for cleaving, but I do know that that's a different type of entry. That's the endocytosis entry, not fusion. So I'm not actually sure. We haven't tested to see how different proteases can affect entry. So I'm honestly not positive, but it's absolutely something worth testing.
Yeah, I think, Bridget, that's a really good point. We also have not really characterized the role of different proteases in the VSV system, but I think that will be important.
Right. So I think there are cell lines where what is TMRPS, that protein is knocked out, so we can see if our viruses are dependent on that or not.
Yeah. So we do have a plan actually in testing these constructs in Vero E6 cell lines, and there's also cell line that was shown that has Vero E6 with overexpression of the TMPRSS2 protease. So that's also something we're testing. But in terms of the cathepsin proteases, we haven't got around to that yet.
Great.
And there's small molecule inhibitors of those.
I have a naive question for the expert here. So along the line of engineering these viruses for a safer testing, can you engineer the system further to recapitulate the other aspect of coronavirus life cycle?
Paul, do you want me to jump in first?
Yes.
So Feng, that's a great question. I thought really hard also with Doug about how we could generate a BSL-2 system, for example, to look at all the processes like replication and translation in the cytoplasm.
Okay.
And because that's really what we are interested in. But it's extremely hard. So the way to do that is you have to set up a replicon system where you basically have, you basically make the truncated RNA genome, right? But it won't have any of the structural gene portion, only the non-structural, the replicase and also other viral proteins portion. The problem is just making that alone, it's gonna be longer than 20 kb. So the problem is it's extremely hard. One way that people have been making infectious clones for COVID is actually PCR amplify each of the individual, so you have many, many fragments, right, over the whole genome, and you amplify each of those by PCR, and then you restriction ligate them, or restriction digest and ligate them together into one single piece. But then every time you will have to make the RNA in vitro and then put it into cells, which is what we do in the lab. But the problem is you're working with something ginormous here. And also synthesizing it in general will be tough. And I think EHS won't be very happy if we put a 20+ kb RNA, which is basically more than 2/3 of the genome, into cells. I feel like they will probably think that's BSL-3 already, just because it's an immediately infectious RNA. So we thought a lot about it, but it's very tricky. And maybe you can make partial replicon system, but then, you know, clearly the virus controls it, the polyprotein production in such an intricate way, right? Where you have the ORF1A, and then only when you have the ribosomal slipping and the ribosomal frame shifting, that's when you make also ORF1B, 1AB together, that it will be really hard to control that ratio. So, and so then the question is how relevant it is. But we would love to have reagents or, you know, BSL-2 systems that we can look at some of the intracellular stages of the viral life cycle, but very tricky with this virus.
Thank you.
Yeah, thank you.
Great. I think we've been some great discussion. Are there any remaining questions? Okay, well then I just want to thank our speakers very much for a really great session, and I look forward to hearing the updates, you guys. It all looks really exciting. Thank you so much for sharing your data today.
Thanks so much.
Melody. Thanks a lot.
Thank you, Melody.
Bye.
Thank you, Dawn.
Bye.
Bye.
Bye.